Well, hello everyone, and welcome back to EV Club. I don't know if you're like me, but I have been sort of just sitting around for the last 8 months waiting for EV Club to start again. And I'm very excited about the event that we're going to have today because we have an exciting paper about EV translation. And thanks very much to Paola de Candia and Ilaria Spatocco, who are in Napoli in Italy. Naples, they're going to be telling us about this scoping review that was recently published in the Journal of Extracellular Vesicles. And I'd like to invite the two of you to please share your screens. And we look forward to hearing what you have to tell us about EV translation. And just to remind everybody, we'll keep you on mute for now, but please put your questions and your comments in the chat box. And then at the end of the presentation, I will allow you to unmute and show your screens as well if you'd like to, and you can engage in a nice conversation with Paola and Ilaria. Go ahead. Thanks so much for joining us today.
Thank you. Good morning. I'm Ilaria Spatocco, and I'm a PhD student in Dr. De Candia's lab. Today, I will present a paper by our group entitled Extracellular Vesicles as Human Therapeutics, a scoping review of the literature. The article has been published in Journal of Extracellular Vesicles on May 13. As all you know, the capacity of extracellular vesicles, or EVs, to target specific cells and to deliver a composite profile of functional molecules have led researchers around the world to hypothesize their potential as therapeutics. While studies about EV treatment in animal models are numerous, their actual clinical benefit in humans has slowly started to be tested. So we decided to evaluate the published literature on EVs as a therapeutic approach in humans to provide a survey on the current state of research. As you will see, the graphical abstract here gives you an immediate flavor of the study. We identify different studies making use of different EV sources and also diversify types of conditions. Our strategy included screening Cochrane and the Prospero databases to search for possible systematic review preparation, finding no entries. Then we searched for relevant articles in the PubMed and the Scopus databases using string which contains terms as exosomes and extracellular vesicles, and therapy or trial or treatment and so on. An example of a string is here reported. Also, the temporal range of the study was from January 1st, 1994 to December 31st, 2023, exactly 30 years. To ensure the minimization of potential error and the correct analysis of the studies, two investigators independently reviewed the identified abstract to determine the eligibility for inclusion in the review. The eligibility criteria for all studies were 1, treating patients with extracellular vesicles or exosomes, and 2, reporting the disease status in patients who received the treatment. Since we expected a smaller or pilot study, we opted for inclusion of publications which report data from an exiguous number of subjects, and no restriction was posed for the comparative treatment nor for the follow-up length. So we also decided to include studies with unclear description of the enrolling criteria and or the methodology used to isolate or characterize EVs. We also decided to not consider studies investigating extracellular vesicles as vaccines because they fall outside the therapeutic approaches category. So since we collected highly heterogeneous studies for different diseases, we soon realized that we will not be able to perform a systematic review, let alone a meta-analysis. So our work fell in the category of a scoping review, which provides a preliminary assessment of the broad nature of the literature available on the topic. A comprehensive analysis of the PubMed database was conducted utilizing the previously outlined search strategy, resulting in about 22,000 records. Then a meticulous screening process led to the exclusion of 8,000 records, categorized as reviews, commentaries, opinions, and so on. The remaining 30,000 studies were screening in details, and all the works about animal studies, in vitro experiment, EVs as biomarkers, or protocols and methods were excluded. Then, upon completion of the screening phase, we identify 40 articles as suitable and pertinent for inclusion in the scoping review. The first thing was to look at the world map, highlighting where the 40 identified studies were coordinated based on the last author's affiliation, showing a widespread distribution, geographic distribution. So then we started characterizing the 40 EV-based clinical studies. In terms of study type, only 8 were randomized clinical trial, while the other 32 were not randomized with distinct design, including 9 case reports. It must be noted that 35 out of 40 studies did not present a control group, and the treatment was compared with the untreated historical group or the other half of the untreated body part. Also, the conditions treated with DVs were highly heterogeneous, as shown in Figure C. So, upon examination of the cellular sources, studies making use of mesenchymal stem cell EVs to achieve immunoregulatory effect were the most represented, 26 out of 40 studies, probably due to their properties as tissue repair and immune suppression of innate and adaptive immune cells. The most common MSC EVs sources were bone marrow, adipose tissue, followed by perinatal tissue as umbilical cord, placental, and Wharton jelly. On the other hand, other peculiar EV sources were dendritic cells, plasma serum, human platelets, endothelial cells, ascites, and also non-human sources, the seaweeds. So in regard of the EV isolation, prior to the clinical application, ultracentrifugation was the most common, either alone or in combination with other techniques. It must be noted that 15 out of 40 studies did not report the EV purification methods at all. While in regard of the EV characterization, the most common was electron microscopy, followed by Western blotting and nanoparticle tracking analysis. Notably, half of the studies used at least 3 independent techniques to characterize EVs, and 16 out of 40 studies did not report the EV characterization methods. So, in our view, underscoring the concerning prevalence of studies that fail to report the employed methodologies, this highlighted the imperative call for standardization of EV isolation and characterization techniques. Among the EV route of administration, the most common was the local one, with 10 studies out of 40, followed by intravenous infusion, intradermal or subcutaneous injection, and inhalation or nebulization. About the 10 studies that use a local administration, 5 of them used a topical one, while the other 5 an injection in another site, for example, wound sites, intraarterial injection, intraarticular, intravitreal, eye drops, or injection into the cochlea, into the mastel cavity, or into the main trunk of the facial nerve. Then we passed at carefully analyzing the 40 articles individually, categorizing them by the treated pathology. The most richest groups were hyperinflammatory conditions, including COVID-19 infection and other chronic conditions, followed by tumor immune therapy, skin diseases, ear and eye pathologies, neurologic diseases, and also 6 studies aimed to investigate the role of EVs in the realm of aesthetic medicine. All the studies, including in the review, aim to use EVs in immune therapy, which aim at strengthening or inducing host anti-tumor immune response. This led to a phase 1 clinical trial. Specifically, epitopes of the melanoma antigen gene were selected to pulse autologous dendritic cells as tumor antigens due to their high specificity. So the metastatic melanoma patients were injected with dendritic cells EVs. Also, a parallel clinical trial used autologous dendritic cells EVs as therapeutics to boost immunity toward non-small cell lung cancer. Another phase 1 clinical trial aimed to investigate the use of ascites-derived exosome called AEX in combination with the granulocyte macrophage colony stimulating factor in the immune therapy of 40 colorectal cancer patients. Notably, though, we not found other studies on the use of EVs in this field. Other two studies aim to ameliorate the graft versus host disease, an inflammatory condition typically occurring after a stem cells or a bone marrow transplant. Specifically, one of the two studies showed good results in one patient, allowing the reduction of steroid dosage. Also, 10 studies have tested different types of EVs formulations as novel interventions to suppress the uncontrolled and excessive immune response and protect COVID-19 patients from systemic organ damage. Infection with severe acute respiratory syndrome coronavirus, the cause of COVID-19 outbreak, can be mild or asymptomatic, but also cause severe and critical pneumonia and acute respiratory distress syndrome affecting the lungs. Interstitial pneumonia is often associated with a massive release of cytokines, the so-called cytokine storm, which is now recognized as a major pathogenic factor for COVID-19, leading to respiratory improvement and acute respiratory distress syndrome. So the first study addressed the safety and efficacy of bone marrow MSC EVs. The product is called Exoflo, as treatment for subjects with COVID-19 ARDS via intravenous injection. Also, a prospective phase 2 study tests the safety and efficacy of Exoflo in reducing mortality of patients with moderate to severe COVID-related ARDS. Both studies showed an increase in free ventilation days, no treatment-related adverse event, and also a decrease in mortality after the injection of Exoflo compared with the placebo. It must be noted that Exoflo is an heterogeneous product which not only contains EVs, but also other proteins and growth factors. Other 3 studies make use of a new developed product named Zofin, which contains EVs from perinatal tissue. One study aimed to assess the safety of Zofin in COVID-19 patients, while another one aimed to treat high-risk patients with mild to moderate acute COVID-19 infection and show improvement in clinical status and also respiratory impairment. The last work test Zofin in long hauler patients with prolonged shortness of breath and respiratory impairment. Also, Zofin is an acellular therapeutic product manufactured to retain hundreds of microRNA, cytokines, chemokines, growth factors, proteins, and EVs. Hence, the described anti-inflammatory properties cannot be univocally reconducted to the sole action of the EV component. While both Exoflo and Zofin were intravenously administered, one study explored the safety of nebulized human adipose-derived MSC EVs in healthy volunteers. The idea of nebulized EVs led to a promising therapeutic strategy for a disease affecting the lungs, such as COVID-19. Also, 2 studies investigate the use of EVs in neurologic disease, as Alzheimer's disease and Bell palsy. Also, a randomized clinical trial tests the use of a blood product called PVRP, that stands for platelet and extracellular vesicles rich plasma, which is known to attain favorable immune homeostatic and regenerative effect. Specifically, they aim to treat chronic postoperative temporal bone cavity inflammation. Another study make use of MSC exosome to treat large and refractory macular holes via intravitreal injection. Finally, a study described the first intracochlear application of EVs in a patient suffering from hearing loss caused by Meniere disease. Specifically, the use of MSC EVs in this condition aim to curb the inflammation typically occurring after the application of an intracochlear implant. And this study show good results, and this certainly paved the way to new studies and new trials. Other 3 studies aim to curb the inflammation of perianal fistulas and ulcers, specifically in patients affected by Crohn's disease. Also, 2 studies aim to contrast the delayed acute wound and chronic wound. Specifically, one of the study used a peculiar chromatographic approach called LEAP, that stands for ligand-based exosome affinity purification, to isolate activated platelet-derived EVs to treat delayed wound healing. They certainly assessed the safety of the treatment, but they did not assess its efficacy, due to the fact that the treatment was injected in healthy volunteers. So they did not observe, obviously, a change in the recovery time. Also, one study published in Turin, Italy, tests autologous serum-derived EVs in combination with the transforming growth factor beta-1 to treat chronic venous ulcers, a condition known to be unresponsive to conventional treatment. Also, a case report tested the use of MSC EVs in a patient with systemic sclerosis, a condition without any approved treatment or gold standard. So the patients showed an improvement only after 1 month from the first dosage. Also, Exoflow, the same product used in COVID-19 as above, was tested in 10 patients with complex regional pain syndrome, with good results, notwithstanding the limitation of the studies of a control lack. Also, other 4 studies make use of MSC EVs from different sources to attempt a therapeutic strategy for dermatological conditions, as facial redness, sensitive skin, and acne scars, respectively, with appreciable effect. Also, 6 studies investigate the use of EVs in the realm of aesthetic medicine. Specifically, one of them tested the potential of EVs from seaweeds, as Sargassum fusiforme and Codium fragile, as cosmetic agents. The EV cream was applied on half of the face, and the other half was used as a placebo. And the study showed an enhance in the skin brightness. The other 3 studies regarded skin rejuvenation. So the last 2 studies test EVs in naturally or chemotherapy-related hair loss, both with good results and as safe as effective. Our comprehensive analysis highlighted the numerous challenges that must be addressed since we could use EVs as a therapeutic approach. Firstly, we need to overcome limitations caused by the absence of placebo controls and use of open-label administration in EV therapy, which both make it challenging to establish a robust proof of efficacy. Indeed, most of the studies were non-randomized clinical trial and did not present a control group. Also, we need to identify optimal cellular sources for specific disease conditions, including non-human sources. There is also a critical need for the standardization of isolation and characterization methods for the scientific community. And in the future, we will also need to standardize the scale-up for EV production. And another issue regards the storage methods, since the freezing procedures has been recently demonstrated to dramatically impact EV integrity, purity, and the molecular phenotype. So we need to develop efficient procedures for storing EVs while maintaining optimal quality, as using fresh preparation is impractical. We also need to identify the most efficient route of administration to minimize EV doses and prevent adverse systemic side effect. Another issue regarded the evaluation of EV efficacy when in combination with ongoing care. Also, we need to establish a dosing regimens to enhance therapeutic effectiveness while avoiding undesired toxicity and serious side effects. We also need to understand the biodistribution and clearance of EVs in vivo, as it is now clear only in animal models. Also, we need to consider the long-term effect of EV treatment. Patients receiving EV therapy should undergo through monitoring for several years to detect any unwanted immunological events or tumor formations. So, as all you know, EVs can shuttle molecules, enhancing the specific targeting and also minimize the off-target effect. However, none of the studies here selected have shown results with modified EVs, and all the published results concern non-engineered EVs. So, the research field of EVs as potential therapeutics has made substantial advancement over the past 2 decennia. However, it is important to consider the findings of the published studies systematically here reviewed as exploratory rather than conclusive. In fact, we observed that all studies are small pilot trials with a large heterogeneity in terms of administration route and target disease. Also, most studies lack a placebo control, apply local EV formulation, and use inconsistent administration dosmetics, making comparison difficult and preventing definitive conclusions about EV safety and efficacy. On the other hand, the recording of some promising outcomes strongly calls out for well-designed larger studies to test EVs as an alternative approach to treat human disease with no or few therapeutic options. Thanks for your attention and the opportunity.
Well, Ilaria, thank you so much. That was very informative. It was also entertaining, a lot more entertaining than some of those awful memes that I showed in the beginning. So congratulations for that too. I have a lot of questions for you. I will, however, keep my mouth shut as much as I can to let other people have a chance to talk. And just as a reminder to everybody, please put those questions and comments in the chat box, and then I will call on you. And at that point, you're welcome to unmute yourself and also show your video if you want. Let me just start though, because I know how this works when you start to engage with the literature and you think you know what you're going to find, but there's always something that comes up that was unexpected. So a question for both Ilaria and Paola, when you went through this process, what jumped out at you as something unexpected, something that you were not anticipating when you started the process of studying the literature?
Well, first of all, Ken, I would say the number. I would expect a higher number of studies to be collected for our study. I didn't expect so many of the aesthetic. I mean, it really weighs a lot. On 40 studies, we have 6 of them on aesthetic medicine. And definitely, it was unexpected to see that not for all studies the EV characterization was in place, or at least it was not reported. This was something that I would not believe, meaning when you are showing a paper in which you have treated people, that was really unexpected for me.
Ilaria, anything to add to that?
I really agree with Paola, because I really did not expect a so high number about aesthetic medicine, and also a lower number of clinical trial in EVs.
Good. So that was something that you were not anticipating. You found that there weren't so many studies. What about this route of administration issue? We've seen some studies where people are doing systemic administration, and others where they're focusing very much on local administration. And if we could just divide that into 2 broad categories.
There
I
was
would always expect that people would do a lot more local administration, because then you don't need to make so many EVs, and you might have a better chance of understanding what they're doing locally. Could you comment on that?
well, yes, there was another thing that that it was actually unexpected to me. I was expecting less local administration. Actually.
Okay.
Yes,
The
yes,
opposite then. Yeah, yeah.
the opposite. But that was my judgment. So I mean, definitely, yes. We've seen a large number in which people are trying to avoid the targeting issue. But another thing that actually was unexpected, was a lot of unexpected things, I have to say, was that none of the studies that we actually found used EVs in which people try to change the targeting of the EVs themselves. So they were used naturally, let's say. And before starting the search, I actually had no idea about that, I have to say.
Yeah, so you were thinking that maybe there would be a lot more engineering to try to tailor the EVs to their target.
Absolutely, yes.
And maybe, I don't know, maybe some of that just has to do with the relative ease of getting a native EV, kind of biologic approved. Not sure. Well, I see that we're getting quite a few questions coming in the chat box right now, so I'd like to hop over there. And so first of all, we have a question here, a technical question about the means of separation. So Ramon, could you please go ahead and ask your question for us?
Yes. Do you guys hear me?
We do. Go ahead. Thank you.
Well, great talk. I really enjoy it. I was surprised, well, should say partially surprised about ultracentrifugation being one of the methods more used. And we have here in our own research have done we work with in vitro cultures, and we have done some mass spectrometry on the centrifugation, and we work with 2% serum supplementation. We collect with EV depleted serum, and even though we have found a large number of protein with only centrifugation as the isolation method in the mass spectrometry, our numbers sometimes go as high as 20% of our top 100%, 100 proteins detected in the mass spec are from bovine origin with ultracentrifugation only. So I was just curious if you guys had any data about the serum supplementation in the cultures where these EVs came from. Thank you.
Well, I guess this is a question for everybody, right? Because Ken, what do you think?
Oh, you're putting me on the spot here. Well, this is the I think there's a broader question of the materials that we use in the culture, but then also all of the surfaces that we're exposing our materials through the production process. And what those additives might do in our experimental system or in the patient. And so, in that regard, it would seem that ultracentrifugation is maybe not the way that you would want to go ultimately, especially if you want to make something that's going to be administered to thousands of patients, because it's, not only is it relatively dirty, but it's also going to be difficult to scale. And so I hear that point, but at the same time, we also know that ultracentrifugation has just been such a workhorse in the EV field, starting with, I think, with virology and, some cell fractionation kinds of things, then moving over to EV studies, that its influence continues to be felt. And I think that a lot of people think about EV separation as something that primarily involves ultracentrifugation, and I think that that's changing now, but it's always been you know, a very popular technique, at least at the research level. All right, so let's move then. There are a couple of questions that I see here about the MSC EVs, right? And so you've, Ilaria, Paola, you found that MSC EVs are very popular. They've been used in quite a few of these trials. And so we have here C. Pinero and also Devrim. Devrim, do you want to go ahead and ask this question then for everyone to hear?
Yeah, Dr. Witwer, thank you so much for this great talk. So my question is a little broad because I'm kind of new in the field. What specific molecules, like particular microRNAs or proteins, are found in these stem cell-derived EVs that can help treat the diseases?
Well, actually, this was another thing that we started to realize while making the study, that this was very unusual to actually characterize on a molecular point of view, not just what the characterization of the EV population, but also the molecular content. If I recall, there was one study about graft versus host disease that was talking about a specific microRNA that could be involved in the action. But other than that, actually what we have found, a few studies were just saying that, as Ilaria said, EVs were not the only component. So that definitely growth factors could be in the preparation or other proteins. And it's just, saying that the effect was not only due to the EVs, or this should be investigated.
So can we say that the beneficial effect of these EVs is just based on their origin right now? I mean, with the information we have known so far?
Definitely for the MSC, I would say so. Yes, definitely. This is growing as a knowledge, as a common knowledge that, the action of EVs coming from MSC are mimicking quite well the action of the MSC cells themselves, I would say. What do you think, Ken?
Absolutely, and that's where a lot of interest has come from. It's there's a range of molecules, including transmembrane and membrane attached proteins that can alter the environment, that can contribute to those anti-inflammatory effects of MSCs. And what I think people had originally shown, well over a decade ago, was that the MSCs that you might put into a tissue, the MSCs themselves might disappear very quickly, but their effects were persisting. And maybe this was, it was found that the MSCs themselves could be replaced by EVs. Maybe some of these EVs are just persisting in the extracellular matrix in these tissues and continuing to promulgate those effects. But again, I think this goes back to the engineering question. If we knew exactly which proteins, exactly which molecules are responsible for that effect, then why not just provide a whole bunch of those in an engineered, maybe an engineered EV, an engineered liposome, something like that. So I think that there's still a little bit of a gap between the outcome of an MSC or an MSC EV experiment and the mechanism that underlies it. And also I guess it's partly the regulatory environment, partly the development environment, where we want to be able to achieve those effects with individual molecules, but maybe we're not quite there yet, and maybe we need to have the EV as a platform, not just one single thing. So definitely some insightful questions here, and then I think they lead us to really put some thought into our strategies. I would now like to move to, actually there's, again, two people who have asked a related question. So Kushi Shukla and Juanma Falcon are both asking about dose. So Kushi, could you please tell us a little bit more about your thinking on this?
Hello, everyone.
Hi there.
Hi, am I audible and visible?
Yes, yes, we can see you. Yes, thank you.
Okay, so I'm a second year PhD scholar, and I'm like new to this field of EVs. And basically, the one thing which I'm more concerned about, the EVs which has been engineered or like surface modified using some chemicals, what are like their current scenario in the patient care? Like, have they used it yet?
Kushi, we just lost you. Could you just repeat that? Just unmute yourself and repeat that.
Hello.
Yes,
Yeah,
go ahead.
so my question is, like all the kind of modification and the engineering that we are doing on the EVs to make it more specific and more targetable to the disease. What is the current scenario of those engineered EVs in the patient care? Like most of the EVs or the like which are in the clinical trials are the normal EVs, which are directly isolated from the cell source. But the scenario in which we can actually use these engineered EVs in the patient.
Well, I think the scenario that we have observed by doing this search is that there is still quite a gap, and this is absolutely expected given it's a young field after all, right, Ken? So there is still a bridge not fully composed between basic and translational and therapeutics. So possibly the engineering we were talking about before will be part of this full bridging that will occur next in future years. What do you think, Ken?
Yeah, and and this is also related to this the question that Juan brought up, and Juan, I'll go over to you in a second here, but when we know what the active ingredient, the mechanism of action is, that might allow us to better dose, right? So Juan, your question was, go ahead.
Yes, my question was how the different works deal with the doses. What is the doses? How they decide the doses? Do they have like a some potency assay that they orientate and then they move, or no? They go directly and let's deal with this amount of vesicles or material. So.
Well, this was very diversified among studies. So first of all, I recall having studying and reading about different modes of quantification, actually. So some people would quantify by protein content, and other by EV numbers. Not all studies were actually clearly talking about the doses that they used, but this is a very crucial point. Very rarely, or in no cases, we have found that the doses used in men or in human was derived from an in vivo mouse model or whatever other animal models. So this was actually, there was again one study about graft versus host disease, in which the researchers, the clinicians were making a computation in terms of number of EVs coming from that number of cells that in vitro would have that type of effect. But there was not a direct correlation between mice and human. Not clear for all the studies.
Yeah, so all these different methods that are used to determine or to measure the dose, I think, are also hampering our understanding of outcomes and of what things are doing. And to go back to what Kushi was asking about, if we engineer an EV based upon our understanding of the mechanism of action, then it's more likely that we're going to have some sort of an assay, like a potency assay or something, where we determine units, where we could very precisely quantitate what the activity of a particular batch of EVs would be, that would get us away from, this, well, did you use NTA? Did you use, whatever method, RPS, whatever it is, to determine your particle number or your protein concentration? all these different techniques are very valuable, but when we're all using different ones, it can make it a little bit more difficult to determine those doses. And in fact, this nicely leads us, I think, into the next question, which is from Hernando Del Portillo. Nando, welcome. Please ask your question here. I think this is a very important one.
Thank you. Ken, nice to see you. Congratulations on coming back to the AV Club. And thank you to Ilaria and Paola for this timely review. I was amazed about the heterogeneity of methods, concentrations, etc. And one thing that if we continue like this, sooner or later we will run into adverse effects of this trial. So I would like to know your opinion, if it is better for the field to continue with this kind of trials, or it will make a harm. Going into a bioengineering, I think it has enormous advantages, but my question is, I don't know if in your experience, continuing with this kind of trials will make harm or good for the field of EVs as therapeutics.
Well, I think this is a very, very, very important question, and I can just give my opinion, but I think that Ken can actually also intervene. Definitely, it seems that we are observing a kind of empirical type of approach right now. Like I'm trying this treatment, and I'm just trying to see what's the effect. Okay. What I really felt when collecting these studies is that people are not really communicating among them. We are not communicating enough. So I think that the thing that we have heard tonight, the new system of like the Ken, help me with that. Like, I think that's really very, very helpful for the future, for kind of organizing studies between people in far apart parts of the world. What do you think, Ken? I mean,
yeah, and Juan, you can feel free to also comment if you'd like, because I think that what we see right now in some sectors is, well, we always have a cyclical nature of capital, for example. So when people who have money, they want to invest in promising companies and promising treatments, and there are ups and downs. And so you have a few companies that fail, and then everybody decides, well, EVs are over. And then a few years later, you get a few good results, and then it goes back up again. And we're, we go through these peaks and troughs from time to time. What we need to ensure is that throughout, whether we're in a peak or in a trough, we're working on the most rigorous or under the most rigorous regimes that we can, to ensure that whether we get positive results or negative results, we can actually believe those results, and that somebody who invests in them can know that this is as good as it's going to get. We've done it the right way. And so that's where, we as an ICEP community, we cannot influence whether somebody is going to invest in a company or not. We cannot tell them to do that or not do it. We can't give that kind of advice, but we can try to create a scientific environment that supports good research and good clinical trials, to make sure that we don't end up with things that fail for the wrong reasons. Juan, I don't know if
if I can add something. So I think ICEF also is doing a good job in that sense, trying to do the effort from the point of view, for example, the task force in the rigorous standardization committee. They have a task force that is led by Eva Rode and Marta, also Montillo. They are trying to be in contact with the people doing these therapeutics, these trials, in order to be able to, in fact, from this, most for example, ExoFlow was one that we were aware in the task force, and we try to contact in order to know more about the products. And it was not easy, because the company or the product was like more like an afraid that we are trying to do an intromission in that, and was not the idea. The idea was to, okay, let's try to communicate, let's try to be together. But this is a not easy point, how to contact the person that are trying to go ahead with the product that they want. And it's not an easy task. But yes, I agree that at some point these trials with the adverse effects, we have to be careful with I mean, we have to aware the people to be careful with that. This is the point.
I guess as a society, if we are already saying about the problems of not using properly controlled potencies, etc., we are somehow covering from an adverse effect that will hamper the field. So I think this is all what we can do. But I'm wondering, if, it's all the times when I read this, are we making more harm or are we making good? But so thank you so much for this timely review, and good to see everybody here again. All right.
Thank you, Nando.
Thanks for the comments. We also have, let's see, Genevieve, you have a question about red blood cell EVs. This is great too. I know we're coming towards the end of the hour here, but maybe we can go a couple of minutes over.
Yeah, sorry, I don't know if you hear me. Yeah,
We do.
I had a couple of questions about but it was answered actually, that there were some publications from companies. So I have been thinking, because I attended two blood EV meeting, and it seems that red blood cells EVs are the big thing. So I was surprised not to find them here. But of course, companies may be doing this and not reporting.
Well, yeah, they are actually growing very much. But I think that our review actually ended at the end of 2023. So there is almost an entire year now. And possibly, this is the idea actually, Ken. We should, in a way, keep going in a few years, right? To have an updated review that possibly will not be a scoping, but a systematic.
Yeah.
We didn't find any red. We could have missed, I don't know, but I think that they are growing a lot in the last, I would say, 12 months. So probably we will see the effect of this growing in the literature in the future.
Yeah, and that's well, I see Genevieve is also saying, what about searching patents? So of course you have the literature, the scientific peer-reviewed literature, but then you also have patents. You have different sources that you can go to understand what's happening across the board. And this dovetails nicely too with Juan's new committee, where there's an interest in understanding the state of the field, the current state of the field, and there are different ways to do that. Literature reviews are one, looking at patents is another. Combining it all would be ideal. Well, we have come to the end of the hour here. I know there's probably much more that we could discuss. We will try to continue to do that in future EV clubs. I just want to say a couple of words about where we're going with EV Club right now. First, to thank you all for your support. And for coming back to this restart of the EV Club today. Over the next few months, what we're going to see is more involvement of some of ISEV's bodies, including the special interest groups most prominently. So you may know that ISEV now has 2 special interest groups. There are more that are probably forming, but they are the interest groups on genitourinary system EVs, or GvHD, and then the EVs in nervous systems. So whether it's the central or the peripheral nervous system EVs, if that's your thing, EVINS is this special interest group for you. These groups are going to be taking 1 EV club per month and alternating months. And then for the other event each month, so I'll probably be running one at the beginning of the month, so the first Wednesday like today. They will be doing the 3rd Wednesday of each month. That leaves a few Wednesdays each month where we could do something else. So we may end up going just keeping it to twice a month, or we might go to a more intensive schedule sometime in the future. But please reach out to me if you have any ideas about what you would like to see featured here. If you would like to present some work or host I'd be very happy to take your comments and suggestions. I saw something in the chat about Discord. Is there a Discord group? Well, as of now, no, this is really something that I mostly do or have done myself in the past. I've had some great help from some volunteers from ISEV. We're going to have more people who are going to be helping with the EV Club in the future, but we'd love to get more people involved. If you have some ideas for setting up a group like that, please reach out. I'd love to talk with you and see what we can do together. So thanks, everybody. I really, really very much appreciate your participation today, and special thanks to Ilaria and to Paola for the nice review that you did here and published in GEV and that you've shared with us today. Thanks also to Juanma for helming this new committee for ISEV. All right, take care, everybody, and we'll see you next time.
Thank you. Thank you.